I think we are right at time, so I'm going to go ahead and have Dr. Palmer to introduce Dr. Su for us, and we'll go ahead and get started.
Great. Thank you, Whitney. So first, welcome everyone to another semester of the MAPme Spring Seminar Series. And this semester, it's my delight to be able to introduce Dr. Jinny Su, who is an assistant professor in the Department of Psychology at Arizona State University. She's a developmental psychologist with training in human development as well as statistical and molecular genetics. And Dr. Su received her bachelor's degree in actual economics at Beijing Normal University in China, and then her master's and doctoral degree in human development and family studies at University of North Carolina, Greensboro. And then later on went to complete her postdoctoral fellowship at Virginia Commonwealth University in 2019. The work that her research actually focuses on how both genes and environment contribute to individual differences in the development of substance use and related behavioral and emotional health outcomes in diverse populations of individuals, particularly as individuals traverse through adolescence and young adulthood. Dr. Su is very, very passionate about ways to increase representation of ethnic and minority individuals and populations in genomics research. And she actually takes cultural genomics approaches to studying the interplay between genetic predispositions and sociocultural factors. And so what Dr. Su will be talking to us today about is the Pathways to College study, which is a longitudinal effort that she began at Arizona State University and focuses on collecting online surveys and DNA from individuals of European American as well as Latin American or Latino heritage. And she's been doing that now at ASU since, if I understand correctly, since 2020. And so the goal of the study is to examine how both family and cultural processes actually work together to influence both substance use behaviors and broader health behaviors. And so today, we're going to be hearing about how her early work looking at the impact of COVID-19 on these students has shed some new light on our understanding of these behaviors. And with that, I'll turn it over to Dr. Su.
Thank you so much for the introduction, Dr. Palmer. And thank you for inviting me to be here today. It's really a big honor for me to have the opportunity to share with you my baby Pathways to College Health study, still a newborn, but I'm excited to see it grow. So I'm gonna, like Dr. Palmer said, I'm gonna share the Pathways to College Health study and some of the new findings from these baseline data from the study on COVID-19 and how it relates to college students' health and well-being. I thought I'd start by talking a little bit about the background, where it all starts. My background, there's a lot of my background, but today I'm going to highlight the work that that is relevant and inspired me to launch the Pathways to College Health study. So like Dr. Pullman said, my doctoral training was in human development and family studies. So the training was really focused on a lot about understanding how social context, right, ranging from family to friendships, neighborhoods, and cultural context, and how that shape adolescent development with a focus on substance use. But it became really clear to me that genetics really play an important role in these outcomes, right? So I was very fortunate to be able to do my postdoctoral mentorship under the mentorship of Dr. Danielle Dick, picture here, in her Edge Lab at Virginia Commonwealth University, where we did a lot of work. I got a lot of training in understanding genetics and how that come together with environment to influence behavioral and emotional health outcomes. One of the projects that particularly inspired me that leads to my launch of the Pathways to College Health study is the Spit for Science study. So some of you may have heard about or read about Spit for Science, but briefly, it's a longitudinal study that focus on understanding how genetic and environmental factors come together to influence behavioral emotional health outcomes among college students. Right, Spit for Science is pretty large in scale. It's partnered with administration at VCU to be able to reach all incoming first-year students at VCU, to invite them to participate in the study. So they have really good participation rate in terms of having about, I think, more than 70% students across cohorts to participate in the study. And so far it has collected, enrolled 5 cohorts of students and with more than 10,000 student participants. So what Spit for Science does is to enroll students during the first semester in college and then follow them up every year, right, throughout college and beyond graduation. And we collected online survey data and also saliva samples for genotyping to be able to understand gene-environment interplay. And it's a large racially ethnic diverse sample. So I was very fortunate as a postdoc working with Spit for Science to be able to use these data to address some interesting questions that inspired me for my current work. A lot of work, like I said, focus on gene-environment interactions. We know that these outcomes that we are interested in are complex, right? It's influenced by our genes and our environments. We know that genes are not our destiny, right? So to me, it's a very exciting and important work is to be able to understand how do various environmental experiences, right, can either exacerbate or buffer our genetic risk predispositions toward developing different outcomes. And so being able to identify these modifiable, right, environmental experiences, I think is very important to offer insights for prevention and intervention. So a lot of my work focused on understanding which environmental factors, right, may play a role in either exacerbating or modifying, buffering genetic effects. So, you know, genetic work over these past decades has really advanced significantly, right? In the sense that we now, it's so much cheaper to genotype. Now we are capable to scan across individuals' genome, right? To look at how across the genome, different genetic variations are associated with different outcomes of interest. So by doing genome-wide association studies, researchers have used large samples to understand how genetic factors are associated with outcomes. Through GWAS, we now know that virtually all of the psychiatric behavioral outcomes that we are interested in are polygenic, right? They are influenced by so many genetic variants across the genome. And GWAS allow us to understand, right, the effect sizes or how each of the single locates or genetic locates across the genome are associated with risk of a certain outcome. Now, to capitalize in advances in genetic research and the findings from GWAS, researchers are able to increasingly commonly use these, what we call genome-wide polygenic score approach, to be able to characterize in individuals' genetic risk predispositions. This approach also captures the fact that these complex behavioral outcomes that we are interested in are influenced by many genes, right? My work primarily used this approach to characterize genetic risk. So I thought I'd provide a brief overview about what this approach is about. So essentially it involves several steps, right? It starts by a GWAS study with a large sample, right? Very large discovery sample. Typically, we're talking about hundreds of thousands or millions that can estimate how each genetic locate, right, across the genome may be associated with a certain outcome, say alcohol dependence, right? So through GWAS, we estimate the effect sizes for each genetic locate And then we're going to apply these findings to our target sample. These oftentimes are smaller sample where we have rich phenotypic measures and environmental measures, right? These can be the Spit for Science, can be my Pathways to College Health study, can be MAPme project, right? And we can then calculate polygenic scores essentially as a weighted composite score, right? That represents how many risk alleles that we carry, right? Weighted by the estimated effect sizes based on the GWAS studies, right? So once we have this polygenic score, we can apply it in our sample to ask interesting questions like how do environmental factors interact with polygenic scores to predict health outcomes, right? So one of the projects that I did when I was a postdoc using data from Spit for Science focused on understanding gene-environment interaction in relation to alcohol use disorder symptoms in a subsample of African-American college students in Spit for Science. I focus on African-Americans because they have been largely underrepresented in genetic research, including gene-environment interaction research. And so I wanted to know whether the gene-environment interaction effects that have been documented to be pretty robust in European populations, whether that can be replicated, right, or generalized to non-European populations. So for this study, I focused on peer deviance. So students report, right, their peers' engagement in deviant behaviors, and also interpersonal traumatic events. So these can include things like experience of childhood maltreatment, sexual assault, etc. Okay. Now, what we found is that, like we would expect and consistent with the literature, right, peer deviance and interpersonal traumatic event both are associated with higher risk for alcohol use disorder symptoms in our African-American student subsample. However, we did not find any significant effect with the polygenic score and also with the gene-environment interaction effects. Now, this finding is really intriguing to us because we thought that can be due to a lot of reasons, right? Could be one of the biggest reasons that we think why we were not able to find the significant genetic effect is actually due to the fact that there's a huge diversity problem in genetic research. That is that the majority of the genetic research, particularly gene environment, GWAS studies, where we identify genetic risk associated with complex outcomes, right? Focus on populations of European ancestry, right? And so with African Americans, if you can look at these graph here, they are highly underrepresented in these gene discovery efforts. And so when we try to calculate polygenic scores, right, applying GWAS findings from these European ancestry GWAS studies to African American samples, there can be a lot of biases, or can be a lot of significant reduction in power. And so this highlights the importance, right, to me, the really importance for us to, one, increase representation of minority populations in genetic research. And I think in genetics, in research overall, not just genetic research, but in research overall, but also to be able to understand the unique pathways of risk for diverse and resilience for diverse populations, and considering factors that may be particularly relevant for non-European populations, or for any population that we choose to focus on, right? So we know very well now, based on past research, that there's substantial differences, right, in terms of prevalence for substance use and mental health outcomes across racial ethnic groups. And there has been documented health disparities across racial ethnic groups, right? For example, we know that racial ethnic minority populations, they on average drink less, right, alcohol, but they are more likely to develop alcohol-related consequences, right, including risk for developing alcohol use disorder. So understanding what leads to these health disparities, right, it's very important. We also know that there can be different environmental exposures and experiences across diverse populations, right? For example, in the United States, racial ethnic minorities oftentimes experience more racial ethnic discrimination, which can be an important stressor, right, that can increase risk for substance use and mental health outcomes. And we really need to look into those factors, those unique experiences for minority populations to better understand what contributes to their risk and resilience. To further complicate this picture, right, genetically speaking, we are also different, right? For example, there's differences in terms of allele frequencies across populations. Some genetic variants that are very common in one population can be quite rare in another population, right? So because of differences across these different levels, I think it's truly important to study diverse populations to better understand their what contributes to their health outcomes. So as a postdoc, I led a spin-off project of Speed for Science where we recruit a subsample of African-American students in Speed for Science, and then asked them to complete an additional survey to ask them about their unique experiences that may be more relevant to them, right? Those factors that were not captured in the original Speed for Science study. So we asked them about things like their experience of discrimination, racial socialization, in terms of, you know, parents and friends, talking to them and conveying messages to them about what it means to be Black African American, their cultural values, right, to instill a sense of pride in being Black African American, etc. And also their racial ethnic identity. We're interested in seeing how these factors may play a role in contributing to their health outcomes. And I'm going to share some findings from this set of study. So with these data, we actually show that yes, racial discrimination actually indeed increased risk for alcohol problems, right? At the same time, we also show that cultural socialization, in this case by peers, for college students, peer really matters, right? Their peers' engagement in cultural socialization with them, like practices that instill a sense of pride in being African American, can actually buffer the risk associated with discrimination. So we see that, when peers' cultural socialization is high, you don't see a negative effect of discrimination. So it buffered the effect of discrimination. In a more recent set of analysis, we also show that racial discrimination actually interacts with impulsivity, which is a genetically influenced trait related to alcohol outcomes, right? They interact to predict alcohol problems in our spin-off subsample of African American students, in the sense that experience of racial discrimination can actually exacerbate the association between impulsivity and alcohol problems, right? With all of that, I think these several studies that I just presented really inspire me and highlight a couple of things. One, I think, is the importance of including increasing representation of minority students in these kind of studies, right? Second is the importance of considering culturally relevant experiences or experiences that are particularly salient, right, or relevant to minority populations, which are largely left unstudied, right, unstudied if we focus on European samples. So that all of that really lead me to launch the Pathways to College Health study at ASU, I wanted to address those two important issues, right? And the overarching goal for this project is really to understand the social, cultural, environmental, and genetic pathways of risk and resilience to students' behavioral and emotional health outcomes. We are really interested in understanding how genetic, right, and both general and culturally relevant risk and protective factors may come together and interact to influence these outcomes. And we are also really interested in understanding whether these pathways of risk and resilience, right, are different or similar across racial and ethnic groups. So I'm sure most of you are aware of this, but I wanted to briefly mention why I think it's really important to focus on college students, right? The transition to college can be with excitement, opportunities, but at the same time, it can also be pretty challenging, right? A lot of students navigating college, leaving home, or forming new social networks and navigating the norms on college campuses can be challenging to them, can be stressful to them. And because of these unique challenges, it's actually been shown that college students have elevated risk of developing alcohol and substance use problems and also mental health problems. At the same time, I also think that college years offers really unique opportunities for prevention and intervention, right? You think about it, this is the, roughly 4 years where we can have relatively easy access to our students, right? To provide service, to engage them in our study, and to feedback to them, to share our research with them, and to engage them in prevention and intervention activities. Because they are on campus, they are in our classes, we can reach them relatively easily compared to after they graduate or if they are not on campus, right? So at ASU, I think we also have a unique opportunity because ASU is one of the largest universities in the United States, and we have a huge and diverse student population, right? So last fall, for example, the first year student enrollment is more than 15,000, and we have really diverse sample with the largest two groups being white and Hispanic Latinx. And that's also why my study, the Pathways to College Health study, are going to focus on these two populations. So here I'm just showing you a timeline that shows the scope of the Pathways to College Health study. We so far have enrolled our cohort 1. This study was launched last fall in October 2020. So it's really, newborn. We are now collecting our first follow-up survey, so time to survey. Data collection is still ongoing. But overall, the plan for this project is really to include several components. One, the biggest component is online surveys for our students. Our plan is to administer online surveys to them once every semester, okay, to capture their other dynamics across the college years. Okay, we also are going to collect saliva samples when in-person data collection becomes feasible, of course. And also, we are planning to also include a component of collecting intensive longitudinal data through daily diary. So these short surveys, 5 to 10 minutes survey, asking students to complete every day across a whole month. We wanted to add this component to be able to, instead of in addition to understanding their behaviors across every semester, we wanted to have a closer look to our students' behaviors on a daily basis. And we wanted to see how gene-environment interactions may influence students' behaviors on a daily basis as well, which has which hasn't been studied that much in the gene environment research literature. So we are very excited about launching that component in the coming fall. We are also planning to enroll cohort 2 in fall 2021. So that's very exciting, but it's gonna be follow similar structure with the cohort 1. So in the next few slides, I'm gonna share with you the key constructs of interest that are included in our online survey, right? First, we are very interested in a broad range of students' outcomes, including alcohol, substance use, right? Risky sexual behavior, mental health, including anxiety, depressive symptoms, loneliness. We also include some measures of positive outcomes, such as life satisfaction. We also include measures on a variety of what we call general environmental factors that have been shown in the literature to be relevant, right, for in predicting students' health outcomes. And many of these factors has been included in gene-environment interaction research with European American samples, and we wanted to look at these in our minority sample as well. So we include things like parenting, peer relationships, romantic partner relationships, social support, stressful events. We also include a measure of family history of substance use, mental health problems. This is an interesting and important measure to us because we know that family history, right, it captures both genetic and environmental pathways, right? And because of previously mentioned potential low predictive power of polygenic scores for non-European populations, our plan is to also use these family history measure as a proxy for predisposition risk or genetic risk in our analysis. Now, of course, when we were planning this study, COVID hit, right? And so we also added some measures about COVID-related experiences. Now, this is a very important part of our project. We included measures that we call culturally relevant risk and protective factors, including, for example, things like racial ethnic discrimination, socialization, their cultural values, acculturation, and ethnic identity. We also include measures and psychological factors or individual characteristics, including things like alcohol expectancies in drinking, because a big focus of my work is on alcohol, and also impulsivity and emotion regulation. So I'm going to describe to you a bit about the sample in our study. Our inclusion criteria is really that students need to be age 18 or older. They self-identify as white or Hispanic Latinx, which are the two largest groups in our student population at ASU. They need to enroll full-time and be first-year college students, right? For our cohort 1, because everything had to occur online, right? In person was difficult at the time and not feasible. So our recruitment strategy all occur online. So we partnered with administration in our college and to have access to contact information, email for all our first-year students, and then we sent them email invitation, right? We also advertise electronically by creating a banner that it's viewable by all ASU students. Because we wanted to reach out to more Latinx students, we also reach out to student organization that's geared toward Latinx students to try to increase our reach to them. And we compensate our students for $15 Amazon gift card for their time. So for our cohort 1, we enroll 425 students. We have about 35% self-identify as Latinx Hispanic. Our sample is predominantly female. We always have more female participations, and I would love to hear any feedback, advice, how to increase participation from our male students. Okay, so the majority, this is a bit surprising to me, a majority of our students report that they actually lived in residence halls even in fall 2020. Okay, of course, with social distancing policies and guidelines at ASU, we have about 30.5% report that they were living off campus with their family. And then the majority take all courses remotely. And then we have significant proportion take at least some in-person courses. With that, now I'd like to transition to share with you a little bit about our recent findings about how COVID may have impacted our students' health. So in our data, we ask how students have experienced, or whether they have experienced, a series of different stressful events related to COVID. So these are things like whether they themselves have been infected, whether their family member have been infected, close friends being infected, whether they or their family or friends have experienced stressful events such as, job loss, financial challenges, or being hospitalized, or family member or friend's death because of COVID. So we can see that significant proportion of our sample, right, 70%, reported at least one stressful event related to COVID. So these experience of COVID-specific stressful event, it's really common in our sample. In addition to experiencing these concrete stressful events, right, our students also report a lot of worries. So we asked them, right, since the onset of COVID, right, how worried are they about their themselves being infected, their family or friend being infected, their physical health, their mental health, right? You can see that we have significant proportion of students reported that they are very or extremely worried about themselves or their family member being infected with COVID-19, though they seem to be more concerned about family and friends. We also have significant proportion reported that they are worried about, very or extremely worried about COVID affecting their physical health and mental health. We see that students seem to be more concerned about their mental health being impacted by COVID-19, okay? Now here I'm showing students' alcohol use behaviors. Here it's data from our sample, Pathways to College Health. We see that the majority report some drinking, right? But overall, drinking level seems to be not very high. Now, how does that compare to non-COVID sample, right? Remember that our cohort is very unique because they were the first cohort of students who started college in the middle of the pandemic, right? So here, I also provide some data from Spit for Science, these also from the first semester in college data, okay? So it's the same timing, but different cohorts. So in Spit for Science, we see similar patterns, right, of distribution in terms of drinking behaviors. This is how frequent they drink in the past year, and this is how many drinks that they drink on the days that they drink. We see pretty similar pattern, although the Spit for Science sample seem to be drinking a little bit slightly more frequently and also a little bit more, but the difference is not huge, I would say. Now, what about students' mental health? Here, I'm showing you just a couple sample items from our measures for depression. This is just one item. you see that the majority of our students report, so about 60% report that yes, they felt depressed at least some of the time, right? And also significant proportion of our students report that they felt isolated from others. So I think this is also consistent with what we have been hearing, right, in the news so far, that a lot of people, not just students, but also our college students are struggling with mental health in the pandemic. Now, this table, I'm showing you some comparisons in terms of means of the key variables in terms of COVID experiences, and also their alcohol use and mental health outcomes between the two groups in our sample. We see that our Hispanic Latinx students, on average, they report that they're more worried, right? They report higher level of COVID-related worries, but they drink less frequently and drink less compared to our white students. There's no difference in terms of mental health, okay? Our Latinx students on average tend to come from lower SES families compared to our white students. Our Latinx students are more likely to be living with family off campus, and then more likely to be taking all classes online, likely due to the more worries as well, right? Now, what we find is that there is really substantial heterogeneity in how our students are doing, okay? So what I'm showing here on this slide is findings from latent profile analysis that try to classify our students into different latent classes based on their patterns of behaviors, in this case, alcohol use and mental health, right? So we have frequency of drinking, quantity of drinking, binge drinking, alcohol use disorder symptoms, depressive symptom, anxiety symptoms, loneliness, and life satisfaction. These scores are all standardized, right? So 0 means the mean level, right? And above 0 or, represents the degree of the standard deviation above the mean, and then below is lower than the mean, right? So we identify 4 latent classes that characterize different profiles of our students' adjustment. The good news is that it seems like the majority of our students are doing pretty well, okay? Despite these challenges, right? So we have 53% of our students being classified as, I call them well-adjusted group, okay? So you see that these gray bars, this group, they report lower than average alcohol use, right? And lower than average mental health, and higher than average life satisfaction. So they're doing pretty well. However, we see a significant proportion, 22% of students reporting having mental health challenges, right? Looking at these blue bars here, right? These group of students, they don't drink that much, but they seem to have pretty high level of mental health challenges, and they are pretty unsatisfied with their life, okay? Next, we have a group of students that engage in alcohol use only, right? Looking at these yellow bars here, they report higher than average drinking, but not seems to be struggling with mental health problems, and they're feeling okay, right? Relatively satisfied, but they're engaged in drinking, okay? Finally, we have a small proportion of our students who I label them as comorbid, right? Who engage in really high level of alcohol use and also seem to have some mental health challenges as well. So these are the orange grouped here. So next we look at how do COVID-19 related stressors, right, play a role in predicting students' likelihood of being classified into these different classes, right, different profiles? Here, we use the well-adjusted group as the reference group. So these coefficients will represent, the likelihood of being in each of these classes relative to being in the well-adjusted group, right? In parentheses, we have odds ratios here, but you can see that experience of COVID-specific stressful events, right, it's associated with higher risk of being classified into any of these latent classes compared to the well-adjusted group. So COVID-19 stress increased risk for alcohol use or mental health or both, right? Very interestingly, these COVID worries, so this is subjective feelings of distress, right, rather than the concrete experience of stressful events. It's only related to higher risk of being in these mental health problems only group, and it's not significantly related to alcohol use, right? I think this kind of makes sense, right? For people who are really worried about being infected with COVID, they're probably less likely to go out and drink, right? Of course, we don't know whether students are the drinking context for our students specifically here, but based on our prior knowledge, right, college students, when they drink, they typically drink in peer context, for example. Next, we wanted to look at whether these effects of COVID-specific stressors, right, hold above and beyond the non-COVID-specific stressful life events. So here, the non-COVID-specific stressful life events, we include a checklist of stressful life events, including, for example, experience of childhood maltreatment, physical assault, sexual assault, etc. Okay, these factors in previous research has been shown to be really robust predictor of these outcomes. And we see that even after, right, considering for these non-COVID specific stressful life events, the effects of COVID stressors still hold, right? It's in similar patterns than what I showed you before. And next, we added more factors, right, to include robust factors across multiple levels, individual factors such as emotion regulation, family factors such as family history, parent-child relationship, and then peer support, and then also cultural values such as familism, right? And even after adding this, we still see effect of COVID stressors, right? We see that the stressful events remain to be significantly predicting increasing risk for alcohol use only in comorbid groups, although the effect for the mental health problems group disappear after accounting for this. But we also see that, like we would expect, right, having a family history of alcohol, mental health problems really significantly increase risk, right? And also having a positive parent-child relationship quality tend to lower risk, particularly for having mental health problems, right? And also folks that have more challenges with emotion regulation seem to be really struggle with mental health problems and also have risk with the comorbid alcohol and mental health problems as well. So with that, I want to summarize. I think these findings, these preliminary findings that we have from our sample really shows that there is substantial heterogeneity, right, in our students' adjustments in their first year of college in the middle of the pandemic. And then COVID-related stressors really are robustly associated with our students' outcomes. And so I think it's really important for us to understand how we can better support our students, right, in view of these stressors. And I think it will be very interesting and important for us to with our longitudinal data follow-ups to see whether the impact of COVID is, just short-term, temporary, or is going to be long-lasting, right? So with that, I would like to end my presentation by acknowledging my students in my lab, the Genes, Environment, and Youth Development Lab here at ASU, and also want to thank ASU and the Institute for Social Science Research for supporting my work. And of course, thank you to our student participants. Without them, the study won't be possible, right? So thank you so much for your time. I have my email listed here. I look forward to chatting with you in the next 15 minutes or so, and also afterwards as well.
Well, thank you, Dr. Su.
Go
ahead, Whitney.
Thank you so much, Dr. Su. I really enjoyed that presentation. So I wanted to make sure that the students have enough time to ask questions. So, we have a pretty small group. If you want to unmute yourself to speak, you can, or if you want to put your question in the chat, you can, and we'll read them out loud. Just let us know your preference, and I can read them for you. I guess in the meantime, though, I do have I have two questions, but I won't ask both of them at the same time. But the first question I have is about your cultural relevancy kind of assessment. And I think that I'm actually starting to dive into this literature myself. And, there's so many scales out here. We talk about, racial and ethnic identity. There's scales on stigma consciousness. There are just different types of ways to reflect, traumatic events, discrimination, and how you then kind of realize that in your everyday lives. What is your process for going about kind of capturing which assessments that you believe would really, capture this kind of space? Do you do some sort of assessment of your student body first? Do you kind of ask questions? Is it just you and a research team kind of diving through the data? How do you go about that process and narrowing down? Because obviously people can't sit there for hours on end. So what goes into that?
Thank you, Whitney. Such a great question. And it's complicated, right? And the cultural experiences, the unique experiences of any cultural group are so nuanced and complex, and it's so hard to capture all of them. And I think the measures that we included in our study can only capture one small piece of it. And to be truly honest, the decision for what measurement to include in our study is primarily based on the literature, focusing, we try to focus on, the measures that has been validated and has been, shown to be robustly associated with the outcomes that we are interested in. In addition, it's also strategic because the minority group I'm focusing here, it's our Hispanic Latinx populations. And I know with the current funding that I have, I can only recruit as not as large as the sample that I have. And I want to be sure to include measures that are parallel, at least to some degree, to other well-established studies with similar scale and topics, so that I can collaborate with them to either replicate or combine sample to harmonize data to do more powerful analysis. So at ASU, we have tremendous expertise in cultural influences with Mexican American and Latinx populations. And I have a colleague, Nancy Gonzalez, who led a study called La Familia, a longitudinal study with Mexican American sample that focus on understanding, cultural influences on substance use and mental health outcomes. And so a lot of my choice of measures are actually, trying to be consistent with what measures that are included there. And because she's such a expert in this area, and also help me, to trust her, right? If she included, and they have published a ton in this area. So these are a couple of decision points that I made. But I hear what you're saying. I think it would be great, ideally, right? We can do particularly when we're studying a culture group that we don't know so much about, it would be great to do mixed methods approach, right? To maybe do focus groups with our students to hear more about what they have to say, right? What are their unique experiences? They are either general, culturally specific. Maybe we miss a ton in the literature, right? We are replicating, ourselves, but maybe we miss a big picture out there. That's possible too.
Thank you so much. That did answer my question. Thank you. That was great. We do have a couple of questions in the chat now, so I will just read the first one aloud. We have one from Katie Martin. She says, "Thank you for your excellent talk. She loved hearing about this project. She's wondering if you expect to see differences in COVID stress or substance use between students living on campus versus off campus."
Very interesting question. We didn't, the analysis that I presented, we didn't look at that, and we haven't looked at that. Though I suspect that we'll have enough power to do that, right? Based on the chart that I showed you, we have 60% live on campus and then others not off campus. The true answer is, I don't know based on the data, but I can speculate. This will be speculating, we all can guess, right? On campus, I know different campus may be different, right? On campus, our ASU has really strict policies. You can live in residence halls, so you can have your autonomy and independence in your space, but you have to follow social distancing, and I think they're not going to check whether you go out and drink or not, but my understanding is, can be totally off here. They have very serious consequences if they are caught, like if they are seen not following guidelines of social distancing, etc. And based on our knowledge of students, college students drinking context, like I said, oftentimes when they drink, right, they drink with peers, they hang out with peers. So I think because of the potential consequences, right, of drinking with and hanging out with peers on campus or close to campus, I would say actually living on campus may actually reduce risk for students to engage in drinking. And there has been literature showing that students' drinking levels on average has declined since COVID hit, during COVID, on average, of course. And that varies a lot, right, depending on where you live, access to alcohol, and your worries about COVID, etc. But in general, due to social distancing restrictions, right, that reduce the risk of opportunity to drink in peer context, and hence that reduce the risk for drinking. Now, living off campus, that means you have more freedom, right? Nobody's gonna, you don't have university-related consequences. You're not gonna be caught. So that allows more room, more freedom. But again, still, I think depending on the stress and the situation, it's hard to say how that may affect their risk.
Thank you. And so we do have another question from Maya Emilio, and she says, "Hi, I am curious about how some of your daily diary data compares to the retrospective reporting in the survey."
Thank you. I'm also interested too, because those data are, we haven't collected those data yet. We plan to collect it in the fall this year. So would be their time 3, right? Wave 3 data collection. Sophomore year, fall semester, we will collect those data. So the answer is, I don't know yet, because we don't have the data yet. But based on my understanding and the literature, right? Daily diary and daily diary measures on daily drinking, right? Can capture very different things compared to the yearly measures or just half-year measures that we ask. How often did you drink in the past year, right? Or how frequent, how many drinks did you drink? etc. Oftentimes in the daily diaries, we see the data being more skewed, because not every student drink every day, but it tends to be more accurate, representing, day-to-day measures. And I see, the utility of having daily measures across a month, right? You can aggregate across, one way that you can use the data is to aggregate their responses across the whole month and to get a more accurate measure of alcohol use in the past month or during this month compared to retrospective measure of use in the past year. So, but yeah, I don't have the data yet, and I would look forward to using that data and see how it can help.
Okay. Thank you. And so we have a question here from Sharon Rabinovitz. I hope I pronounced that correctly. She says, thank you for your important work. How is this data informing the work to ensure universities can support their students coming back in the fall? I know this is a very broad and layered answer, but would love to hear your thoughts.
So important question. And, this is why we do this work, right? We want to ultimately be able to serve our students. Right? One of the reasons that we wanted to do these person-centered analysis to identify, to understand the heterogeneity in students' well-being, is that we are interested in seeing, how students are doing, how are they different, what if there is problem, what is the major problem, right? Is it drinking? Is it mental health, right? What is it? If universities need to focus on something, what should it be? I think our findings shows that it seems to me, that's my take, is that it seems to me the mental health challenges is the major challenges for our students compared to alcohol use. We know that at least on our campuses and a lot of efforts pre-COVID cohorts, right? A lot of orientation efforts, for example, or prevention efforts focus on preventing alcohol use because we know that's a big thing. But it seems to me for this cohort in particular, mental health challenges, it's really a major issue. And I think universities, as they prepare to invite students back on campus, I think more focus should dedicate on providing mental health services to our students and recognizing that, these challenges could be long lasting, right? Mental health challenges. So being able to actively send out messages, right? To start conversation about mental health with our students and making sure that services are readily accessible to our students can be really important. Focusing on mental health more than alcohol use, I would say, for this year.
Thank you. And I know we're right at time, but we have a couple of more questions that have come in the chat, and I want to respect your time, Dr. Sue. Will you have a little time to answer these last two? Okay.
Yes, of course.
Okay. And so this question is from Megan Yang, and they ask, what kind of differences might be observed in adopted children or children of varying immigrant backgrounds?
I wanted to clarify the caption, question. Adopted children versus children with varying immigrant backgrounds. Okay.
Right.
Can you clarify adopted children? You mean adopted from a different cultural background or?
Yeah, so they would definitely have like a cultural disconnect with both the one they were adopted into and then the one that they left.
Yes. Yeah. Well, that's a very interesting question, and I think these can be can vary, right, depending on a lot of things. The cultural connection, the cultural background, and also the quality of relationship of the adopted child with their parents, the family, right, and how much acculturation stress that they may experience, right. We know that for immigrant immigrant families in particular, and actually a lot of the students in my sample are immigrants, children from immigrant families as well. In these kind of families, we know that acculturative stress can be a very important factor, right? The degree to which they feel the stress stemming from wanting to be acculturated with the mainstream culture, and at the same time also enculturated to their own heritage culture. And this tension, right? This stress can oftentimes, and has shown in the literature, be a risk factor that can increase, mental health challenges and drinking to cope with the stress. Now, you mean with varying immigrant background, and also generation status is also an important factor, right? The immigrant families literature has shown these interesting paradox, right? For example, for alcohol, the second, third generation, they tend to drink more to a similar level with white students, and so it's really nuance. I think there's no simple answer about how they might differ. I guess the question would be, it depends on their generation status, the stress that they experience related to acculturation, the family relationship, etc. Because these outcomes that we are interested in are so complex. They are multifactorial, right? Influenced by genes and different factors across different levels. So, yeah.
Thank you. And our last question comes from Lauren Burton, and she asks, she starts with, she really appreciates you sharing your work with us. Her question is pretty specific. She is very interested in the interaction you spoke about between racial discrimination and impulsivity. I noticed that you had looked at negative urgency. Did you look at other domains of impulsivity as well? And if so, what were your findings?
Excellent question. Yes, we use the UPPSP scale to measure impulsivity. So that captures 5 dimensions, right? Negative urgency, positive urgency. I know these terms may sound really weak, sensation seeking, and 2 others. In terms of the interactions, we found that these interactions primarily it's with negative urgency. So experience of racial discrimination exacerbates the association between negative urgency and alcohol problems. So negative urgency is about, I tend to drink or act impulsively when I feel bad, right? And so it's about these being impulsive and engage in risk-taking behaviors when experiencing negative emotionality. And I think this interaction make sense, right? Experiencing discrimination can really increase the level of emotionality, negative emotionality, and that can exacerbate these risks.
So thank you. We have some more comments in the chat. If you get a chance to read that in your time before you get off. We thank you so much for coming out to speak with us, Dr. Su. I'm sure you can tell by our interesting chat that everybody is engaged and really enjoyed your talk and all the work that you're doing so far. And I want to just turn it over to Dr. Palmer to see if he has any last remarks for everyone.
Well, yeah, I just want to echo the sentiments that were just made. It was a fantastic talk, Dr. Su, and from MAPme to you, we look forward in collaborating as well. We ask very similar questions, and so I think between ourselves, Spit for Science, I think students would be very, very fascinated to see, all their hard work really leading to change. I'm not sure if anyone realized, but actually Dr. Vorbitovitz was actually the administrator for Emory's college student health promotion. So it's really great to sort of, be able to reach out and make these kinds of connections and see that that's also happening at Arizona State as well.
Yeah, thank you so much. It's really a pleasure, and yes, I agree that we do have a lot of shared interest, and we share our goals, right, with MAPme, and I look forward to learning so much more from you all.
Yeah.
And our future collaboration.
Great. So I think with that, we'll wrap up this spring's event. And as always, this event will be available on our lab website channel as well as our YouTube channel. So if you've missed it, this will be a great venue to come and see us. Take care, everyone.
Thank you. Take care, everyone. Bye.
Thank you, Dr. Sue. That was great. Really fascinating work. Thank you.